start the record. Okay, so now that tells me the recording is going. I'm going to, I've got to share the screen here. And then I'm going to load up the PowerPoint. And so please, okay, buddy, load. I guess I missed something here. Do that. So did the PowerPoint show up?
Yes.
Yes, it
Okay.
did.
Okay. All right. So I guess we're,
Earl,
yes.
Just quick before you get started here, I have several people who have asked to make sure that they will receive the recorded version, and we are assuming that this is being recorded for their purposes.
Perfect. So if the recording is on right now, so we'll save it and Janie will forward it to everybody or upload it into the shared drive for use. Okay. So I guess I was going to say my name is Earl Holzapfel, but I think almost everybody on here knows who I am. And I think I've already explained the mechanics of how we're going to do this. So we certainly welcome you to the Milestone and Investor Virtual Summit hosted by myself, Ralph Parchment, who's a co-founder, inventor and current chief scientific advisor, and just an amazing guy. He's the guy, the reason we're sitting here, and David Schaefer, who manages our investor relations and business development. So welcome to all of you who have been investors. Hopefully others will be joining you soon, and we'll be speaking to that more in a moment. But as I've been joking around with all of you to start off with, the early investors, there's just nothing like what you did for us, and we're deeply indebted to you because you really got the ball rolling. And I had introduced a second ago Marion Gibbons, who was the first investor. So congratulations to you, Marion, for getting the ball or the snowball coming down the hill. As all of you know, we developed this unique nanoparticle delivery platform to maximize the bioavailability of the famous anti-cancer agent fenretinide. And Ralph, in a minute, is going to describe what bioavailability really means and why it's important for drug use and functionality. And that work led to our lead product, ST001 nanofenretinide. So we're here to discuss the progress and status of that endeavor. We're going to break this down into 4 parts. My further comments by Ralph on the science and why the science is so relevant to the commercial field that we're headed into. David Schaefer will be talking about our commercial status, our fundraising status, and what the future looks like. And then we'll have a Q&A, which we hope we can make a lot of fun. Here in front of you is the required safe harbor statement. Obviously, you all know what that is. So what I'd like to do is, talk about first the most important milestones that have been achieved, really starting back when we finally got patent approval and then move on from there. So you all know that we're a clinical stage specialty pharmaceutical company, and the nanoparticle is our claim to fame, and ST001 is what's taking us there. But this drug, it's unique in the sense that there's amazing history behind it, and that's what sort of builds the foundation for what happened with our involvement in this. Prior to just going up this step, a diagram, I just want to remind you of the history real quickly. When Ralph Parchment, myself, and Dr. Al-Khatib were at the Kamanos Cancer Institute, we were approached by the National Cancer Institute to solve the bioavailability problem with this drug, fenretinide, that was under study. And Ralph's going to talk about that in more detail. So we took on the challenge, formed SciTech, applied for grant funding, and received various levels of grant funding over a bunch of years. We also got National Institutes of Health commercialization assistance program support, and that brought in Luz Garmuzos, who's been with us ever since then as one of our key people, other than his short sabbatical. So now let's look at this slide. So the first and most important thing we did was, after proving the concept of what we had, was putting a patent together that would be accepted by the international reviewers in the United States. It was not a trivial matter because of the complexity and diversity of the delivery system, the mechanisms of action, the fields of use, the composition of matter, and so on. But we got that done. Obviously, that led us then into the step of what are we going to do with this commercially, and that's what Ralph Parchment and Dr. Al-Khatib and Lou got involved in, filing our IND application to the Food and Drug Administration. And I can tell you the job that Ayad and Ralph did in crafting the protocol, which really is the template for the clinical trial plan. It was extraordinary because it was unusual. It was fast or accelerated. We left out portions of it, and Ralph will describe that later, that are normally required in order to increase the speed to approval. And we had been told by a lot of regulatory affairs experts outside of our organization that the FDA would never approve our drug, but it did happen. So that was a really important milestone. About the same time, we got what's called orphan drug designation, which is a designation given to folks who are investing in rare diseases. And from that, you get extra market protection, and so that was really important to us. And you also get tax rebates covering some of the cost of the development of the drug. We then moved into 2020, prepared to commence into the clinical trial. We came out of the JP Morgan Healthcare Conference and expected to be proceeding forward fast. Then the pandemic hit. During that same period of time, we realized that our drug had significant potential in the treatment of viruses. So we put a whole hypothesis together for doing that, filed a provisional patent application with the patent office, and started putting together a IND application at the same time. And out of that whole exercise, came an opportunity that still exists. But being an organization of our size, we had to focus on oncology. So as soon as we were able to jump back in on that, once the clinical sites were ready to go, that's where we focused. So the next step after the provisional patent application and the filing of materials with the US government was IRB, Investigational Review Board approval from Rusk University Medical Center in Chicago, where our principal investigator, Dr. Timothy Gazelle, who's head of hematology, oncology, and cell therapy, agreed to serve as the PI, and Dr. Al-Khatib recruited him. So we're really lucky to have an organization like that in place. So now here we are at the present time, and we now have 2 INDs. We have one approved, and the second one that we submitted 2 weeks ago. So we're anticipating the results of that. But we are clinic ready, okay? We have been clinic ready in oncology for some time, but we're more prepared because we've done a better job of accomplishing other tasks that I will speak to in the moment and next, okay? So the next challenge was completing specific tasks that you wouldn't call them milestones, but moving forward. One of the things we kept asking ourselves is this drug that has so much potential with so many different cancer indications and other diseases potentially. Is it too good to be true? And, someone asked the question, is that possible? One of the people who was on the call with us this morning said, well, think about it this way. Fenretinide was a 1970s invention and problem, and we arrived at the solution in the last few years. So we have got an elegant solution to an amazing drug shortcoming, and that's really our claim to fame. But there is that time frame that you have to take into consideration, where people could be skeptical about whether this drug was really going to achieve the potential that the studies, so many studies show we will realize. So we have to be cognizant of that as we point out that this is an amazing drug, and we have solved the problem with a really 21st century invention. And I think as Ralph goes through it in a few moments, you'll know what I'm talking about. So let's just talk about what we accomplished specifically in the last few months. We optimized the clinical development plan and budget. Why? Because there's so many off ramps, on ramps, depending on which drug, which disease you're testing, how far you're going in the IND process with a particular indication. And there are lots of variables for how that might get funded. So we had to have a plan that took all that into consideration. And with a couple of clicks of a button, we can change our budget and see what the costs are. And we can show investors with, exceptional detail exactly what will cost, where the costs will lie, and they're really believable. They're very complex budgets. We also needed to, add and expand to the team. So I'm going to jump to the next slide for a second. So on the science and technology side, the people you see, Lou Scarmuzzo's, Ali Moyen, Michael Young, and Ralph have been incredible players. Ayad Al-Khatib, who was our chief medical officer, was instrumental, but a great contributor. So we've had that area covered with exceptional talent. We've added to the team by bringing in professionals in finance, investor relations, legal, and so on to come full circle in making sure we've got all those aspects, all aspects of business development covered as well. David Schaefer brings in investor relations and fundraising experience from his investment banking days, where he's raised more money than I can even imagine. I can't count that high. I don't know what the numbers are, but we're exceptionally lucky to have him with us. And then on the business development, strategic partnerships, PR and marketing, the people at the bottom have joined us. And so we're now operating, with a team that's expanded from when most of you originally got involved with us. We've got the team to take us through the clinical trial, and we will alter that team over time as we move into commercialization activity. We validated the clinical interest and market that started years ago with Dr. Al-Khatib and myself, and going to various events, and Michael Young, but we've expanded that through the involvement of the other people who are shown on that chart, talking to pharmaceutical companies, investors, end users, and so on, to make sure that this drug has a market. And it's, we don't have a drug looking for a market, so to speak. We got the IRB approval at Rush, and we hope to have a similar approval from Spectrum Health in Grand Rapids for the COVID application in the near future as well. We have the second IND that we filed on the 12th of June, thanks to Bob Patterson and Lou and Ralph, and we're looking forward to hearing back on that any day. So that's exciting. We have confirmed without a shadow of a doubt that this drug has significant potential as a combination therapy by going back and looking at the research that was done by others using various delivery systems and looking at those results, seeing what the shortcomings were of those drugs, the strength, going back to those pharmaceutical companies and saying, don't you want to collaborate with us so we can extend the patent life of your product or improve the response performance in clinical trials or whatever. So we are really sitting in a fantastic position to move forward. So in conclusion, it's sort of giving you an update. During a time of international stress, we are poised to quickly move into the clinic to confirm the new fenretinide's promise. And I say new because it is a new chemical entity. It is a new drug. The energy that is emanating from discussions with potential funders or collaborators is absolutely palatable. We've not experienced that kind of response as we are now. Historically, a lot of interest, but now it's really excitement. So at this point, what I'd like to do is turn this over to our co-founder, inventor, Ralph Parchment, to tell you about the importance of fenretinide and its relevance in the clinic.
Thank you, Earl. Good afternoon, everyone. Thank you for joining today's investor update. This part of the presentation is focused on the science behind our lead product, ST-001. And I'll briefly discuss three scientific topics that are relevant today. The first is fenretinide's bioavailability problem that's hampered its previous clinical development by other organizations. Second is the significant clinical and commercial upside from solving this bioavailability problem. And third, the novel and patented technology that SciTech has developed that actually solves the bioavailability problem so that fenretinide can return back to the clinic and be properly developed to treat cancers that, from clinical trials, we already know respond to this drug. So I'd like to begin by explaining the term bioavailability. You'll see it throughout this presentation. Earl's already alluded to fenretinide's problem to be solved, which is a bioavailability problem. So the definition of bioavailability is simply the percent of an administered dose that enters the bloodstream. So you could start by understanding that with intravenous therapy, where a drug is injected directly into the bloodstream, that has a bioavailability of 100%, all of the dose. If the infusion is done correctly, all of the dose enters the bloodstream. And then if you move to other routes of drug administration, like by mouth or the oral route, only a fraction of the drug is usually absorbed into the body and makes it into the bloodstream. So oral dosing typically has bioavailability less than 1. And that's going to be important in understanding the fenretinide bioavailability problem. So I was a co-investigator on the first clinical trial of fenretinide to treat cancer in adult patients. It was conducted at Wayne State University's Karmanos Cancer Institute. And my role on that trial was to analyze the fenretinide blood levels in the treated patients and try to describe the relationship between the doses that they were receiving and the blood levels. One would expect that as doses are increased, blood levels of the drug should also increase. What we actually discovered from that analysis was that above a certain dose level, as shown in the slide, around 1,800 milligrams per day per body surface area, we reached a plateau on the absorption of the drug. So increasing the dose did not further increase the blood level of the drug. And that's illustrated by the chart in the right-hand side. This chart also reflects what was happening in clinical development with fenretinide in the pediatric cancer population, where the investigators running the first phase 1 trial in children with cancer, neuroblastoma in this case, encountered the very same phenomenon. So if you look at the chart on the right, the blue line represents the blood level, which is shown on the y-axis as a function of dose on the x-axis. So as the numbers on the x-axis get bigger and bigger, that indicates higher and higher doses. And what you're seeing here is that that curve starts to go up in the beginning at low dose levels, but as the doses approach 1,000 and beyond, it starts to flatten out. Ideally, we'd be on that dashed red line. That dashed red line is an indicator of a twofold increase in blood level of drug when the dose is increased by twofold. It's proportionality. It should make sense that if you double the dose, you get twice as much drug delivered to the blood. But that wasn't the case with fenretinide. Doubling the dose, quadrupling the dose resulted in a very small increase in blood level. So that is the bioavailability problem, and that is the problem that our lead product, ST-001, solves. So that's important to understand. So the bioavailability problem solved by ST-001 occurs because ST-001 not only is an intravenous product, so it can be injected directly in the bloodstream, but it also delivers very high doses, much higher than can be delivered by mouth, effective doses by mouth. So can you go back to the slide before, please?
Oh, I'm sorry.
So not only do we want to be on that red dashed line, but with ST-001 and intravenous dosing, we're going to push well above that red line. So we're going to realize the full potential of fenretinide at the highest possible drug levels, much higher than can be achieved with the oral dosing. So usually a phase 1 trial results in a recommended dose for phase 2 development, phase 2 trials, that's based on the safety and tolerability of a drug in the phase 1 trials. For the first time, though, in our professional careers at our phase 1 team at KCI, at Camaradas Cancer Institute, had to recommend a phase 2 clinical trial dose that was based on the maximum number of capsules that a patient could swallow in a day. And I put a picture of these gel capsules. They're quite large. We ended up recommending 30 capsules per day to be swallowed by these patients in order to achieve the maximum possible blood level from oral dosing. So despite the fact that the maximum tolerated dose of fenretinide was never reached, and that the recommended phase 2 doses was just based on the number of capsules that could be swallowed by patients in a sort of a practical recommended dose. That recommendation was evaluated in several phase 2 trials, which I'm going to present next. And those phase 2 trial results show the upside and the potential upside of solving the bioavailability problem. So if you go to the next slide. So one such trial took our recommended 30 capsules per day and used it to treat ovarian cancer patients with recurrent disease. And in the orange box on the bottom right, you see a very important result from this trial. So in the patients where blood levels exceeded 9 micromolar, which is a concentration measurement, there was an 18-month overall survival of 66%. However, in the patients who had lower plasma levels that were below 9, the 18-month overall survival was only 13%. So that dramatic and statistically significant difference proves the importance of raising blood level as much as possible in order to achieve maximum clinical response rates. And this was not the only trial that showed that. In the next slide, I show data from the pediatric neuroblastoma trial I talked about. I've divided their data into 3 groups. The dose levels are shown over on the right-hand side, and they're color-coded to the blood levels on the chart on the left. The 3 different dose level groupings that I've made differ in the blood levels that they cause, shown in the chart. What you can see on the right-hand side is that the response rate, SD is stable disease. So you can see I had 22 patients in the yellow, 20 achieved stable disease. At the next lower dose level above that, 400 to 700, only 12 of 16 stable disease. And then as it fell to 100 to 300, in that grouping, only 9 of 15. So you're seeing a declining disease response rate as blood levels are declining. So despite the bioavailability problem and the inadequacy of those gel caps that the patients were taking, you can still see the potential upside in raising blood concentrations of fenretinide. And you can imagine the clinical activity if we're up on that red line or we surge past that red line with ST-001. So I put in the bottom right-hand square there just some quotes from the investigators of this trial. Maximum tolerated dose was not reached. It again was the practical issue of just how many of these gel caps could a small child swallow. In many cases, the doctors had to remove the oil from the gel cap and try to mix it in milk or something in order to dose these children. It did not influence lipid or triglyceride profile, and toxicity was not dose limiting. So there's tremendous upside to increasing blood levels, and there's tremendous room to do so because fenretinide was not toxic at these blood levels that were achievable with the gel caps by mouth. Next slide. And then recently there was an intravenous formulation that was tested in T-cell non-Hodgkin's lymphoma patients with escalating dose. And this trial showed the same thing. It showed the significant upside to higher blood levels and the improved clinical response of patients treated at those higher levels. In the chart, I've shown 4 dose levels from 640 up to 1,810 and the number of patients in each group. At the 640 level, there were only 2 of 5 patients that showed just stable disease. But as you reach 905 and the higher dose levels, you start to see complete responses, partial responses, etc. Now, unfortunately, what I showed in red that happened in this trial is that every dose level that was associated with clinical activity was also associated with severe dose limiting toxicity. So it wasn't possible to find an effective dose that was well tolerated for treating the patients. However, the potential of fenretinide in this disease, I think, is really illustrated well by the case on the left-hand side. This is a young woman, 30-year-old woman who enrolled in the trial. And you'll notice in the footnotes there in red below the case that she had received and had not responded well at all to prior chemotherapy, 2 regimens of chemotherapy, as well as a FDA-approved retinoid called bexarotene that's approved for cutaneous T-cell lymphoma. That was her disease. And then she also went on 2 trials of one for an investigational immunotoxin and the other for another class of drugs called HDAC inhibitors, which subsequently have been approved by the FDA to treat cutaneous T-cell lymphoma like this woman was diagnosed with. So despite all of those previous FDA-approved regimens failing her, she went on the fenretinide trial, and within 6 cycles, the disease cleared. You can see the dramatic impact of fenretinide on her disease. On the left-hand side is before she started treatment. You can see nodules of T-cell lymphoma in her skin, throughout her body, in this case in the pictures on her face and head and hands. And then after 6 cycles and by the end of her treatment, when the doctors decided to stop treating her because she was in complete remission and there wasn't any need to continue treatment, you can see the disease had completely cleared. So unfortunately, this formulation, despite it being intravenous, is going to be difficult to use because you can't separate severe toxicity from the clinical activity. If we go to the next slide, I just wanted to summarize what you've seen in terms of formulations and how they compare to ST01. So in all of these examples, starting with the gel caps that we tested at Wayne State in the very first clinical trial, through a product shown in orange called Limixorb, which I didn't talk about because it really was not that significant in advance. Limixorb is a fatty acid formulation. It's kind of drinkable fat, so to speak. The fenretinide was dissolved in that and patients were asked to drink that. It did result in about a 2-fold increase in blood levels, as I show there, from 20 up to about 42, but not significant enough. And many of the patients dropped out and did not want to continue to take the drug that way. And then in the green line, I show a summary of the blood levels from the trial I showed in the previous slide in CTCL. That IV formulation did dramatically increase blood levels. You can see it raised it from 20 in the blue up to 240. So about a 12-fold increase in blood level probably explains why there were complete remissions on this trial in T-cell non-Hodgkin's lymphoma. But note the composition of that emulsion. That was an emulsion that was built around soybean oil, which is laced with triglycerides. Its actually primary component is triglyceride. And you all know that triglycerides are not healthy for you, so you can imagine what happens to your body when you infuse these directly into your blood system. The commonality in these 3 products is that they're all off-the-shelf vehicles. They weren't developed around the fenretinide molecule. They were not developed with fenretinide's properties in mind. They were just available, and fenretinide was stuck into them and found to be acceptable for trial use, and they went to trial. So it was speed over appropriateness. It's kind of like, in my mind, kind of like buying garden tools to fix your car and then wondering why it's taking so long to fix the car. So anyway, if you go to the next slide. So all three of these off-the-shelf vehicles, although certainly fast into the clinic for clinical trials because they're already available, all carry significant liabilities that limit their use. All the way from the bottom there in the blue bar where I talked about how many capsules per day had to be swallowed by the patients and the bioavailability problem that oral dosing creates, through that orange limit sort of formulation that still is administered by mouth, still encounters the bioavailability problem of drug absorption from the gut, and lack of patient compliance. It's a miserable thing to, not yet, Earl, go back.
Yep, sorry. Yeah.
And then finally in the green, because the emulsion is a low strength emulsion that's got a very high content of triglyceride, there's several liabilities to that in addition to the toxicity. One is that it requires 5 days of hospitalization for constant infusion. So because it's low strength, it takes a very long time, a long infusion time to get in the appropriate dose. So it's very expensive, it requires inpatient care, and it's very tough on patients because they have to receive an IV infusion for 5 days straight, 120 hours straight. There's triglyceride toxicity, obviously, because of direct infusion of triglycerides. It also contained egg phospholipids, so patients who have egg allergies were excluded from getting it. And it requires a test dose for patient eligibility. So the low dose is the test dose on day one, requires a stat lab, medical lab test to make sure patients will be able to handle the triglyceride, the high triglyceride dose. And only those patients who pass that laboratory test are eligible for the treatment. So about 1/3 to half of the patients will fall off. So what we tried to do with STO1 is to design a formulation sort of with an engineering approach. We call it formulation by design. So not only did we want to avoid all of those liabilities of those previous products, but we also wanted to incorporate some design elements, so to speak, that were very desirable. So in this orange box on the right-hand side of the slide, you can see some of those design goals. And like I say, this is very similar to an engineering approach where you assess the problem, you introduce design goals that you will adhere to, and then you incorporate those design goals into the prototype. And that's exactly what we did. So a couple of the really important ones is that our phenretinide nanoparticle system, exemplified by STO1, can reach very high strengths in the formulation. Our patent claims cover 6 to 75 milligram per milliliter, which is 3 to 37-fold higher than the emulsion that's shown in this green row on the left-hand side. That was 2 milligram per cc. STO1 is 12, representing a 6-fold increase. And we know we have prototypes in the laboratory for patenting purposes that get up to 60 and 70. So we have a tremendous range of phenretinide doses that we can administer. Because we are high strength, it minimizes the infusion time. So our treatment would be outpatient based. The patient can come in, receive a 4-hour infusion and leave the outpatient clinic and come back the next day. It does not require inpatient hospitalization or being attached to an IV pole for 5 days in a row. We avoided triglycerides. It's completely triglyceride free. In fact, we restricted our ingredients to those that have clinical track records of safety, thinking that it would help our IND chances at the FDA to use only ingredients for which there was prior experience in approved or investigational products. And that actually turned out to be the case. Our product doesn't require test dosing. Therefore, we think it'll be tolerated by all patients who are treated. And finally, the Limixorb, which is the orange line on the left-hand side, contains some lipids that are biologically active. And we wanted to avoid those as well because we did not want to introduce a second biological activity in our formulation. So in the next slide, just to summarize the problem in our R&D goals and how we developed STO1. So remember that the problem is that we need to increase the bioavailability. So we accomplished that by designing an intravenous product. We also wanted to design a safe product with the potential to far exceed achievable blood levels so far. Remember that there hasn't been any phenretinide attributable toxicity so far in these trials. It's all been formulation toxicity. So we think there's tremendous upside in going into very high blood levels of this drug. So how do we solve the problem? So we developed a formulation that mimics cell membranes, and those cell membranes are made up of chemicals called phospholipids. Those phospholipids self-assemble into what are called bilayers. So there's 2 layers of phospholipids, and I'll show a picture in a minute, but they're kind of oriented tail to tail. When we mix phospholipids with fenretinide and put it through a manufacturing process that creates nanoparticles, it yields nanoparticles like are shown in that picture on the top right. That's an electron micrograph of 120 nanometer diameter particle. What you see is a clear, a whitish particle, but it has very dark lines, concentric lines, kind of like cutting an onion and looking at the cross section. And each of those dark lines is a phospholipid bilayer. So you can imagine that if fenretinide likes to exist in the phospholipid bilayer, and phospholipid bilayers have very high loading capacity for fenretinide, having 50, 60 of those within a given particle creates very high payload capacity for fenretinide. And that's why we're able to achieve such very high concentrations and formulation strengths. So regarding discovery and preclinical development, we formulated with membrane phospholipids, like I said, emphasizing those with clinical safety records to not create concerns at the FDA about our IND. We're using hypoallergenic ingredients, so we're avoiding things like egg products. So all patients, no patients would be excluded from allergies. And we were able to develop a scalable process that's suitable for manufacturing all the way from trial material through commercial material. And as Earl mentioned, this R&D program was funded by peer-reviewed SBIR grants, as well as some bootstrapping from some outside activities that Earl and I were conducting. Next slide. So how do these particles work? So again, the picture, the electron micrograph in the upper left shows the actual structure, and then to diagram that structure, you begin on the left-hand side of this path. You see those gray molecules with the red circles. Those red circles are the ends of the phospholipids that like the water, so to speak, and the gray are like the fatty parts of the molecule. So they self-assemble and make what's called a bilayer, which means these 2 molecules of phospholipid tail to tail, so to speak, and that's what surrounds a cell. That's what makes a cell membrane. And fenretinide, as a retinoid, likes to exist between those lipid tails, those gray parts of the molecule. So if you replicate the membrane structure over and over again into the layers of the onion, you get that purple structure on the right, where those red lines indicate, or similar to the black lines in the photograph, they indicate individual bilayer membranes, all loaded with fenretinide. So after the IV infusion, these particles circulate in the body, they are deposited in the tumor area, and they bind to the surface of tumor cells. The tumor internalizes them, metabolizes those natural phospholipids, releasing the fenretinide, and that fenretinide then moves to the nucleus as well as some other molecular targets to exert its action. And I just want to reemphasize that if you remember the T-cell non-Hodgkin's lymphoma patient who had been treated with a retinoid, an FDA-approved retinoid, and had not responded, but still responded to fenretinide, indicates that fenretinide not only acts as a retinoid, but has additional mechanisms of action, which is probably why it's so active in cancers. There were also cases in the neuroblastoma trial where patients were resistant to FDA-approved retinoic acids for the disease and went on to respond to fenretinide. So at the bottom there, our conclusion is that fenretinide affects multiple biochemical pathways leading to the death of the cancer cell, and will be useful even in cases that are resistant to the FDA-approved retinoids. Next slide, please. So the outcomes of this R&D program, representing where we are now, we have a patent issued for the fenretinide-containing phospholipid nanoparticles. And like I said, we can achieve drug strengths up to 36 times higher than any of our competitors. We also, using this technology, we are also improving fenretinide's natural solubility in water, so to speak, which is kind of like blood, by a factor of 75,000. So far and away, the best way to formulate fenretinide and make it suitable for intravenous injection. The IND was accepted by the FDA, and there's the IND number. And as Earl alluded to, we were able to have our IND accepted by the FDA without performing any animal toxicology studies. And the reason I think for that is that we put a design constraint on ourselves to only use ingredients in this formulation that had clinical track records, known safety profiles. So the FDA agreed with our argument in our IND that we did not need to perform toxicology studies because there was no toxicity information that needed to be generated. It's already all known from past clinical experience. Like I said, we have a manufacturing process and a manufacturing partner ready to produce trial material. Our protocol for T-cell non-Hodgkin's lymphoma uses an accelerated design to rapidly reach biologically effective doses, and then we expand to more patients. That guarantees that fewer patients will be undertreated and more patients will receive potentially therapeutic doses. And then by design in our phase 1 trial, we're including expansion cohorts after we've determined the recommended dose in 2 types of T-cell non-Hodgkin's lymphoma, which are the two T-cell diseases for which fenretinide has already been proven to be active in the clinic from that emulsion trial that showed the toxicity liability. It was very important in showing that those 2 diseases are responsive to fenretinide. So next slide, Earl. So in summary, so we think fenretinide is a valuable asset because it's been formulated in ST001, and that removes or eliminates all the liabilities of the previous formulations that have stalled the development of fenretinide multiple times by multiple organizations. We believe that there's a large underserved market in the oncology space. I showed you 3 trials where there was clinical activity in 3 different cancers despite suboptimal administration of the drug. You can imagine if we can increase blood levels by 10 or 20 fold, what the outcomes in those diseases might be. And furthermore, this seems to be a delivery platform. It should be able to benefit other stalled drugs with similar properties, drugs that need to be formulated in these phospholipid environments and protected from water in order to administer them intravenously. So thank you for your attention. I wonder if there's any questions on science.
Why don't we save it until we heard from David? I've heard us talk about this technology over and over again. I've never seen it as well presented as Ralph did at this event. So congratulations, Ralph. It's really a lot clearer as to what is unique about this product. David Schaefer is now going to talk to us about our commercial activity, etc. So it's all up to you, David.
Thank you, Earl. Thank you, Ralph. And thank you, everybody, for joining us today. It's with your generous support of, early investors that the company has successfully been able to raise in cash grants and materials almost $6 million to date, which has enabled SciTech to gain the IND for ST001 and also to be prepared to enter the clinic as Ralph has described. By the way, I'm the guy who's going to ask you for money. Aside from that, I'm really excited to be a part of this effort. In my 30 years working with early stage life science companies and in raising capital, I've rarely come across ideas and opportunities that have the market potential that I see in SciTech and ST001. And Earl, to your point, fenretinide is so 1970s. ST001 is the drug of the 2020s in the future. You've heard Earl and Ralph refer to ST001 nanofenretinide as a novel new drug with just huge potential. They covered the drug's current status, its vast potential, its unique capability to achieve therapeutic bioavailability, its therapeutic breadth, and what I think is one of the most understated important items here, its affordability. Oncology as a market is looking for safe, really safe and affordable breakthrough therapies. And ST001 really could be that breakthrough. The market opportunity for SciTech and ST001, as you can see in the slide, is very large, initially targeting roughly $2 billion for T-cell lymphomas and small cell lung cancer indications. The market potential could reach $25 billion as additional cancer indications are added, such as breast and ovarian, and several others. And as the nanoparticle delivery platform expands to include other water-soluble drugs, water-insoluble drugs, that type of number becomes a lot more visible. As you can see, we expect ST-001 to address large markets, several of which are presently underserved or designated with unmet need, meaning there's no other medication currently in the marketplace that serves that particular indication. Once the safety profile of the new formulation has been validated, the opportunity expands to include several oncology and viral indications, many of which we think can be monetized via strategic license. We also believe, relative to most other new oncology drugs that are currently being evaluated, that ST-001 will not only be very safe and effective, but will provide a simple, safe administration regimen and also be much more affordable than these other new and prospective treatments that we see costing well into the hundreds of thousands of dollars. Slide 2, please. The clinical trials that you see designed and in front of you are designed to confirm that ST-001 solves the bioavailability challenges that have confronted fenretinide historically. It's time to talk money. We expect to enter the clinic shortly following a successful tiered Series A capital commitment of $20 million. The tiered raise is milestone-based. What does that mean? It means you commit for the full program, but the funds are allocated only after each trial's success. We think that works for you. It also works for us. It works for all of us together, as with each stage, the company is further de-risked. Additionally, an investment of only $3 million, 3 of the 20, will enable SciTech to commence and complete the accelerated portion of the phase 1A safety trial, as you can see in the first column. That's the first measurable milestone. Our clinical trial plan, the IND, has already been approved by the FDA. It's important to note, and I really want to emphasize this, that these phase 1 safety trials are confirmatory. They're not exploratory. And that there's already a significant amount of data from past fenretinide trials in over 3,000 patients. As Ralph indicated, toward the end of the phase 1A accelerated dose escalation study, we would expect to see signs of activity in patients. Successful completion of the 1A study portion opens up several pathways forward. And that is really exciting because this is not a one drug, one indication company. This is not a one drug, two indication company. This is not a one platform, one indication company. This is a one platform, multiple potential drugs, multiple potential indication company. And that sets the stage for not only the revenue that we talked about, but also for significant end market and exit valuation. The key attributes of these trials are as follows. It's an accelerated design, as we indicated already with FDA approval. The trial is not lengthy. Each segment is it's just 4 to 8 months. The patient populations required to complete these segments are small. Phase 1A accelerated is just 8 patients. And has been noted over and over again, and I think really important, for you all to understand, is that the time to market here, from the time we commence the study to the NDA filing and ultimate approval, should be less than 24 months. I point that out because the typical cost of bringing new oncology drugs to market today is over $1 billion, and that can take a decade or more. We believe that SciTech can accomplish this goal with your $20 million investment in less than 24 months, based upon substantial FDA guidance that we've already received. We believe that the trial will also confirm that SC001 can be used as both a single agent and a co-administered combination therapeutic. Independent published papers over several decades support fenretinide's potential as a standalone treatment for several cancer types. Once again, that drives the end market. It also drives valuation. Additionally, recent lab research conducted at Texas Tech University points out fenretinide's effectiveness as a safe and effective co-therapeutic. Also, an additional value driver for the company. Next slide, please. So why are we asking you to invest in this important effort? There's several significant reasons. We feel SciTech is a unique opportunity for all the reasons that you've heard today. To start, ST001 is clinic ready. The IND is in hand. We have an FDA orphan drug designation. ST001 has FDA guidance to rapid commercialization following a phase 1b trial. ST001 is already significantly de-risked. We've talked about the fact that it's been trialed in several thousand patients to date, and we have data that has enabled the company to bypass several arduous FDA steps in these trials. ST001 targets, as we pointed out, large addressable markets and significant unmet needs. And the oncology market really needs that. It's a very important step that I think can't be overstated. Finally, we think that ST001 will have significant treatment advantages over both existing and future treatments, including safety, efficacy, broad applicability, and affordability. This is about changing people's lives. And we encourage you to have a follow-up conversation with Ralph to gain additional insight and confidence in the science, to gain a better understanding of that. As I mentioned, we're looking to raise a tiered $20 million Series A round to accomplish these goals and position the company to make a significant difference in the lives of cancer patients, advancing ST001 as a safe, effective, and affordable oncology treatment. We're asking you to join us in this important mission by investing in us, investing in this team, investing in this asset, investing in SciTech and ST001. We would like to thank our existing investors and invite you to add to your stake. For those of you who are new to the SciTech story, we invite you to join us. I'd now like to Earl, I'm going to turn it back over to you. We can open the floor for questions.
Well, thank you.
Thank you all very much.
Thank you very much. You can see the kind of talent that's been brought to bear on this, and we're very excited that you could share in this. So with that, it's time to ask questions, make comments. And so the floor is open either to talk or to send a chat message. So who'd like to start? Somebody?
Earl, I have a two-part question that came up in chat. It is, how much is left on your convertible notes? And the second part of the question is, what are the terms of the convertible notes?
Okay, so we had started off with a $1 million convertible note, and we increased it to about 1.25. So we've got about 400,000 remaining on that note. But if there is significant interest in that vehicle initially before moving into Series A, we can expand it. And just have to go back to the SEC and notify them and get others who have invested already to agree that that makes sense. Secondly, the other part of the question is, the investment or the conversion in the convertible note is based on a 25% discount to the value of the units or shares that a Series A investor acquires, or it can be calculated at a maximum value of the company of 17.5 million. Probably at this point in time, it's that number that will be a greater discount than the 25%, and then we have an interest rate of 6%. So that it's an interesting way to go. Whether we will be raising more money in the convertible note round or not is really a function of the type of investors, their level of interest, their availability to invest. And we're open to that, as well as just going straight to the Series A.
Thank you.
How about somebody else?
Yep, there's a scientific question, Earl, I can take. The question is, how specific is the take up by cancer cells, and why would normal cells be spared? Yeah, good question. So the research by us and many others published in the literature says that fenretinide exploits abnormalities in cancer cells that are actually vulnerabilities to fenretinide. And so the drug does circulate in the body. It would be found in different tissues at different levels based on their blood flow, the ability to pump the drug out of the cells, et cetera. But it seems like fenretinide is targeting a number of critical pathways that don't function very well in cancer cells, and therefore are create that liability. So I don't know if whoever's asking the questions, you want to follow up with something in more detail, or is that the answer? Is that a good answer?
Yeah, I don't know if you can hear me. Thanks.
Yes, yes.
Yeah, I appreciate that. But what happens when you start looking at other, I guess they're more lipid-based drugs, non-water solubles
Mhm.
down the road?
Yes.
You know, how likely is it that you'll find some that are non-toxic to normal cells?
Yeah. So I guess maybe I could rephrase it. I guess you're sort of getting at whether there's any tissue selectivity to the delivery or uptake of the particles.
Yeah, well, that was kind of the basis
Yeah.
for my question. It sounds like
Yeah.
it's not that specific, but which is what I suspected. But so it's more, it's a great delivery system for non-water soluble molecules
Mm-hmm.
that intercalate into the, the bilayers.
Right.
And how have you looked at what those molecules might be, relative to their toxicity profile?
Right. Right. So in our tissue distribution studies in rodent, this is in rodent models with tumor, there are tissues that show selective delivery and especially selective retention of fenretinide delivered by the particles. It's very hard to know if that's because the particles are still residing there, or if that's because the particles delivered the fenretinide, they were metabolized and the fenretinide released. There is a distinction there, so I can't tell you it's particle associated fenretinide. But where we did show in several tissues and tumor is that the retention of fenretinide is enhanced by formulating in the particles. So whether that would extrapolate to other drugs, I'm not sure. If you're asking, does it take a lousy drug and convert it into a winner because it can selectively target cancer? I think the answer to that's no. It's not going to save a lousy drug, but it is going to, I think, restart development of drugs that are stalled because they can't be administered any other way. That there have been attempts to administer them intravenously, and it's been associated with liabilities that are not due to the drug. It's rather due to liabilities from the formulation. And those are perfect candidates in this situation. For example, other retinoids that have stalled out in development over the years, and like you mentioned, other membrane active agents, maybe even outside of oncology, and other therapeutic indications, for example. I don't know. Is that getting to the point of your question, or?
Oh yeah.
Okay.
I mean, I don't want to sort of push you to give me other examples of specific drugs, but I'd be very
Yeah.
curious. But whatever you want to answer is fine. Thanks.
Sure. Now, just one other point. So for the engineering approach, we call it formulation by design. It includes consideration of the size of the molecule and the space that it occupies. That's part of the patent. So each drug candidate for formulation, would have its own pocket of space, so to speak. And the idea is to fill around it with phospholipids of various structures, while still holding ourselves to the constraint that there has to be a clinical track record, ideally, in dealing with the FDA, a safety track record, and that they're naturally occurring. We don't want to get into lipid structures that are artificial and might be biologically active and introduce an unknown variable, into the formulation. So those would be the constraints.
All right, well, thank you, Ralph. Those are well articulated responses. And I know, we certainly, David, have thoughts on other drugs and so on, but maybe we could hold discussions with you offline if you're interested in pursuing that further, because clearly, we've had meetings identifying target drugs, but we didn't want to lose our focus on using ST001 and attacking the early indications to confirm in a clinical setting that this is truly what it's represented to be from prior data. Any other questions? Dr. Al-Khatib, you have one there?
Yes, first I wanted to thank you for arranging this update, because I think it's important for us to know where you are. So I really appreciate the effort, and it's wonderful presentations. If you can update us on the scaling up and the production of manufacturing process, and where do you stand with that? Because you have the trial ready, you have the IND approval, you have the PI lined up at Rush, which I hope still in effect or in force. So you need the drug. Remember when I first chatted with you, I told you to do a clinical trial, we need three things. You need the drug, you need the IND approval, and you need the money. So where do you stand with the manufacturing and the scaling up of the production? Because once you start the clinical trial, you would want to make sure that you keep going.
Well, as you probably recall, the FDA, prior to giving us the final approval on our IND, required that we scale the manufacturing to some degree. We did that at Campbell, but then they were as concerned about making a sterile product and making a reproducible product. So that portion we accomplished, and then we have moved on to an organization called Plow Center for Sterile Drug Production. And maybe Ralph, you're a better person to describe where we're at with that. But Harry Kachet of that organization was on with us this morning. I wish you'd been here then, and he could have told you why they're the ideal party to move forward. But they're sitting with inventory ready to go. We have a contract all but signed in place, ready to make the drug. And so in that case, the only thing we're really missing is the third point you raised, Ralph, Ayad, which is the money. So Ralph, why don't you finish answering it?
Yeah, to add a couple things. So specifically, we've transferred the manufacturing equipment from Campbell to UT Plow, ready to go. And like Earl said, the inventory of materials is there. And one thing I'd like everybody on the phone call to know is that in addition to the NIH support of the R&D effort through SBIR funding, the National Cancer Institute has also donated the active pharmaceutical ingredient fenretinide from their supply to use in our formulations for these cancer trials. So we do not have to manufacture or qualify active pharmaceutical ingredient or API. It's being donated from the NCI supply. So that really does kickstart the manufacturing and offsets a significant number of costs that would normally occur in having to manufacture API. That it's we have it available. So I guess I don't know anything else to add. I other than say that they're all set, ready to go. We've done a tech transfer familiarization with UT Plow. I walked through the manufacturing master batch record and CMC plan to control quality and the release testing. And They're all familiar with that. It's certainly within their capability. They certified that they have the scale to manufacture clinical trial material for both phase 1 and 2 clinical development. So if you match that with what Earl and David were talking about, accelerated path to market and meeting with the FDA to use a phase 2 trial for market approval, potentially, we might go all the way from phase 1 to market approval just with UT Plow and not have to move to a larger scale commercial manufacturing until we have market approval. So that would be an advantage, that we only would have this one tech transfer step, and then we would stay with our UT Plow partner for the next 2 to 3 years. I think that's an advantage. Does that answer your question?
Yes, thank you. Thanks.
Okay, sure.
Great question, Dr. Al-Khatib. We appreciate it. And we recall your concern about that as we all shared it. So, and it's important to note that manufacturing a product like this is not trivial. I mean, if you wonder if manufacturing is trivial, just look at what J&J is going through with their vaccine right now. The company in Maryland that's supposed to be making it hasn't been successfully doing so. So, or at least it hasn't been approved for use. So that this is something we've worked very hard on. Harry Catcher to Plow this morning, shared in subsequent phone calls after the event that he's really enthusiastic about encouraging his institution to take a potentially greater role in working with this on, going forward and considering some of the other delivery systems that we're working on as well. So it's kind of exciting.
There are
Oh,
no
Marion, yes.
Well, on this manufacturing side, the ingredients that go into this product are easy to get. They're all not hard. I mean, they're easy to get. Someone's not going to corner the market.
Yes. So Marion, yeah, it's a good question. They're US based providers. They come from a place called Alabaster, Alabama. These are ingredients used in multiple formulations of other products as well. And so they are produced at GMP quality and ready to distribute. There's some lead time. Obviously, you can't just show up at the door and say, hey, I need 15 pounds of this stuff. But generally, the lead time is about 2 to 3 months to fulfill an order for these materials. And there's no supply issue in terms of international boundaries or the inability to dock a ship in Los Angeles port, anything like that. They're all US based company. They synthesize them, they qualify them, and they have a track record at the FDA. So we don't foresee any problem with those ingredients.
Okay, well, let me go on. I don't know how I can ask the right question, but if you had an example, because you've said that it's inexpensive to make. So if you were to have a dose for some disease, can you project what a low price would be?
Well, let me try that one.
Yeah.
As part of that budgetary process we talked about, Lou, you're not on here, I guess, or Andrew. So we had come up with sort of an early expected cost of goods once we got up into a commercial volume of something on the order of $10,000 for cost to run a full therapeutic regimen, and that regimen could be many months of therapy. So relative to the other retinoids that are out there, such as bexarotene, which is now costing for the same kind of regimen about $60,000 to $80,000. Or if you go into some of the new CAR T therapies or targeted therapies, it's hundreds of thousands of dollars. This is going to be something cost effective. And then, probably a guy who could give us insight on it, probably won't at this point, would be a guy like Steve Monk, who was in the manufacturing business. But over time, as the volume increases, I'm sure that cost would come down.
Okay.
Phil, Marion, keep in mind, my brother-in-law just went through an immunotherapy treatment segment,
Mhm.
and it was over $100,000 per treatment. And he withstood 4 to 6 treatments. So we're talking about, and you know, a difference magnitude of difference in pricing for a SciTech regime.
And note, I was comparing cost of goods to selling price. That's not really totally fair. But you know, so you can assume if your cost of goods is $10,000, you know, unless you're, you know, a crook, it's not going to be in the range that Bill was just talking about. Okay, so, you know, now I know, you know, it's what the market will bear in a lot of businesses, but I think we have committed to being responsible suppliers of drug to meet the needs on a massive basis. And that's our goal. Just like we're interested not only in therapies for adults, but very much interested in therapies for kids, for pediatric indications. And although we didn't talk much about that, you know, that's a high priority on our list as soon as we confirm the safety of the drug in the new formulation. So we want to be responsible citizens.
Earl, if you don't mind, it's Michael, and Marion, it's nice to meet you. Thank you so much for your help to date. I wanted to just let you know, obviously we are early in the development process here, phase 1, phase 2. It's hard to tell exactly what will end up being a commercial valuation for a product at this point. Most pharmaceutical companies aren't entertaining any of that kind of discussion. And you can see that we're trying to be a bit more transparent here because we believe we have a very viable and useful therapy, in particular for a patient population that I've worked with for over 20 years now. And, you know, I think that there would be a welcome addition for a nanofan retinide, but also certainly something that would be responsibly priced. But yeah, I think it's difficult at this point knowing what our initial cost of goods look like when we get to full scale up, what we understand a full, if needed, a full phase 3 trial would cost, et cetera. It may have moderations or implications for the actual ending price. And we must not discount the fact that should we get into a collaborative arrangement and or an acquisition scenario, those individuals involved in that will have a significant impact on what the outcome price would be. So you understand what the spirit of this is. I just I think I'd be I think we'd be out of line to try and pin that down at this point.
No, I understand. My husband has leukemia and he takes a drug called Jakafi. And it retailed, I think, at 14,000 for a month.
Right.
And
Mhm.
then it came down, the insurance picked up some of it. We get into the deep hole. And so now we just pay $780 a month. But it, you know, some of this stuff is just horrible. I don't know how people get it.
Right. No, and certainly the JAK-STAT drugs are in a different category than what we're talking about here. This is a small molecule approach with a very unique drug delivery system that we have proprietarily developed. And we believe that there will be other future applications here, as was brought up earlier. But yeah, I wish your husband all the best.
Yeah, thank you very much.
Let me just jump on for a second if I could. We're joined here by two people who are outside of our organization, but have a very significant understanding of our technology and the commercial opportunity here. One is Ken Massey, who used to be with Pfizer and then a small private equity company, and Bill Werkmeister, who's with Oncology Impact Partners, is also affiliated with the National Foundation for Cancer Research. So I'm not, if either of you would sort of like to make any comments or whatever that, you know, might be of interest to the audience, why that would be appreciated.
Bill, you need
Bill-
to turn on
Bill,
your.
you're muted. You're
Bill.
muted, Bill.
Oh, am I? Okay, can everyone hear me now?
Yeah. Oh, he's got a
well-
SciTech background. Oh my God.
Well, I figured I would throw up a SciTech background, even though I'm not really with, with SciTech, but our organization, the National Foundation for Cancer Research, is an investor in SciTech, and I run one of our venture funds, our new venture fund that we're trying to launch, OIP Partners, which is focused on clinical stage assets. But I caught part of the talk, Earl, and not part of the talk because we had to put the baby in the bed. And I'm covering a couple stickers that the baby put on me before I put her to bed. But I would just, I would say to the investors that, we thought this is a really compelling investment opportunity because, there's a tremendous amount of data that's been collected over the years on fenretinide, which I think, you had spoken about earlier in the presentation. And, really the core issue that SciTech had to solve in this case was really the bioavailability issue, which they've resolved. I think the other thing that was really compelling to us, at the foundation was, the accelerated pathway for approval for the SciTech asset. So this is something that could go from, trials to approval in, a matter of 2 years, 3 years or less. So just a little kind of background on our foundation, why we got involved with this. And I think, the management team is really world-class, and Earl's been really working on, I think, building it out to make sure that, we have people on the finance side and people on the business side, as well as just the science, which, is the earlier part of the team. So I think that's my summary comment in 1 or 2 minutes, Earl.
Well, thank you. That was elegant. I wish I could talk like that.
I don't know. I think it really takes what I saw some of what you did, but not all of it because I had to put the baby to bed. But hopefully it takes the hour and wraps it down into 1 or 2 minutes. That's kind of what we saw as interest of the company.
Yeah. Ken, did you have anything to add?
Sure. I'm happy to jump in there, and I'm not going to add all that much more other than I've obviously been very involved in and interested in the investment with SciTech since I was with the equity firm. In fact, we had made the decision that we were going to go forward with it. It was shifted a little bit just because the major family whose money it was decided to go in a slightly different direction. But at that time, and I've stayed in contact with SciTech and been just really enjoying the very logical development process that has been going on, and I'm very excited about the direction this is going. And actually realized that while we have ST001 is the main focus of the discussion here, that this is a platform that could actually be used in a number of other applications where there's bioavailability issues. And that's one of the things I think that is, while it wasn't necessarily touched on during the discussion, it probably should almost logically follow. But I think that the opportunity expands beyond ST001, but we want to move towards that mark first.
Yeah, and if I could, I just to address that, because I think it's a really important point to look at this in stages. There's with today's stage, which is the importance of getting ST001 into the clinic to evaluate and confirm the safety that we've seen and exhibited in other fenretinide trials. Beyond that, once we complete that phase 1A trial, this could conceivably go in a number of different directions. So, assuming we get the right investor response post a successful 1A and 1B trial, there could be a substantial amount of money at that point raised and specifically earmarked for identifying those assets that would be, well suited to be combined with this nanoparticle phospholipid delivery platform that Ralph has developed. That by itself is an enormous opportunity, and that's really what moves this from a $2 billion addressable market up toward that larger $25 billion number that you saw earlier on the slide. So what we have to remember here is that, particularly with your help and all of your help and where we've gotten today, is that we have to act like we have, limited resources, and that every dollar we have is allocated effectively. So and you can't really do phase 1B before you get to 1A. So we have to start at 1A, take that to the next level, and have people bear witness to what it is this drug can do, the safety profile, the the pending improvement efficacy, and then as investors and advocacy groups for different cancers see the results and become more interested, that's when this thing could just truly take on several lives. And at that point, the infrastructure of the company would be much larger. All these things are totally dependent on a successful completion of the phase 1A trial. That is so pivotal to the success of this company, and that's why we're here today.
Mhm. Absolutely.
Well put. So are there other questions? We'll open it back up to anybody else who would like to make a comment or ask a question or whatever. You've stayed on longer than we had anticipated, and we really appreciate it, but we're willing to sit here and keep going if you are. So if there are other topics you'd like to bring up or questions you'd like to ask, feel free to do so. Anybody?
Well, on
Well,
the question, how are you? Okay, you're looking for money. So how are you advertising that, so to speak? How are you presenting what you're going to do to give, if someone wants to put in $50,000, is that too little? And then what, I guess I'm just looking for, what are you selling? What do you get for whatever money you put in?
From an
well,
investment. Go ahead, Earl.
no, I mean, I think that, we're flexible depending on the amount of money that will be put in, okay? So, David spoke to that $3 million required to really complete the accelerated portion of phase 1A, which is really what that is really doing is confirming that the new formulation is safe based on the dose levels that we think we have to get to achieve therapeutic response. So that $3 million is important, and we'll take investments in that any way we can get it, obviously. In terms of the major raise, that's in David's bailiwick, and so I think I'll let him answer that. But better rather than me just speaking about that early money.
So, Marion, I think it's really it's not just an interesting question. It's the question, and every company that is in SciTech's position, will tell you that they've got the greatest asset developed since sliced bread. What encouraged me to join this company and what I bought into was the science. It was really understanding the problem that Ralph and his team were trying to overcome. What was it, Ralph? Almost 8 years it took you to develop the delivery platform for fenretinide. This was not an easy thing to do. It's not easy to replicate. And so I point out, if you think about what makes a business successful, it's not just a good product, it's not just great people, but it's having something that's defendable. It's having something that's difficult to replicate. It's having something that addresses a large market. And if we're selling something to an investor, it's all of those things. It's all those things that attracted me to this company. And I first met Earl over 2 years ago. And I watched the development. I saw the pain and suffering. I saw the opportunity. I also had a full-time job. Since then, I retired after a successful 30-year career. And, you know, we were brought on by Earl, my team, to work in investor relations and business development and really maybe take a look backwards and see, you know, if the message was being articulated properly. And what we all agreed upon was that the key element here to advancing this was to focus on the one thing that really mattered, and that was solving the bioavailability problem. It wasn't finretinide. Yeah, it was solving the problem that finretinide was unable by itself to overcome. And believe me, there have been a lot of smart people that have tried to develop their own delivery mechanisms for finretinide. And as Ralph pointed out, every one of them has a big X over them. They all failed. And that was really, you know, part of the hope and the dream and the expectation here that could, in essence, be overcome. And the data that I saw, once again, that just helped me cross that bridge personally, was the fact that it showed that that finretinide, even with some of these other delivery systems, showed great effectiveness. It was just it was the the toxicity, it was the side effects that brought those those particular efforts to a halt. And believing what Ralph has presented, and which is the solution to a problem of a drug that shows so much promise, both in its breadth, its safety, its efficacy, all of that. That's what I bought into, and in turn, that's what that's the message and the story that and the opportunity that we're selling to new and existing investors. Does that help, Marion? You're on
Marion,
mute.
you're on mute.
Marion, just to answer the question a little more directly, we're reaching out to VCs, individual investors, and strategic partners on this. We have a number of people that are willing to put in large scale money if we bring a strategic partner to the table. So we have in the last month and a half, 2 months, put together very direct presentations for these strategics. And we are also speaking to VCs in the area in Michigan, along with around the country with people. And we are just banging the drum on how novel and unique and how potentially life-saving this therapeutic is.
Right. I'm really, did someone write a check and just, that's it? I mean, the actual mechanics of investing. Like my nephew said, if I wanted to do this, how do I do it?
Well, we hope they do it like you did. Just write a check and drop it in the mail.
Write a check and Earl will come pick it up.
Earl will go see your nephew, which is the nephew I know. I would be happy to visit him. And honestly, we've still got the convertible note round in play for small investors who would like to jump in right now. It would take us about milliseconds to put the paperwork together and send it off, so that the check could be sent in with the document signed, unlike what we did with you. Larger investors, obviously, there's a negotiation process. It's a due diligence process. And, we're planning for that. Phil, who was just talking a second ago, has been working with us aggressively to prepare for evaluation by pharmaceutical companies or large venture capital firms or whatever. So we got a whole process. And then there are other team members here are working with social media to get the word out on us. So we are creating quite a new presence on Twitter and LinkedIn. And so if anybody is using those tools, they're all of a sudden hearing a lot from us. And Lori, who's sitting here, has been doing extensive outreach on many fronts. So this is a full court press by a lot of people to get the word out. So giving you the answer, there isn't a simple answer. Laura,
Okay,
you looked like you were trying to say something.
well, no, if you're gonna
Yeah,
go.
I just said it's been a full court press since we've been on board and really trying to tell the story. And you know, one of the things that we, you know, run into is that, you know, there's all these new like groovy drugs. I call them groovy drugs. I come from 20 years in pharma, and but at the end of the day, no matter what people think of pharmaceutical companies, they really do want to help patients, and they have venture funds and all those kind of things. And we've, you know, Lou and I have been working really hard too on the strategic planning on, you know, co-therapeutics and other lipid, you know, insoluble drugs and or water insoluble drugs that can match with this. I mean, we have done extensive work in that arena. So, you know, if we can get one of the strategics, you know, in line, that would be, you know, phenomenal. And we're marching down that path pretty hard. So we're at this every day. And I'm so thankful that, you know, everybody's gotten on this call. This is not going to be the first, and it's not going to be the last. So we're, you know, we're gonna do this, and we're very excited about it.
Okay,
Thank
well,
you.
I'm just saying, if you're at the cocktail party, you start talking, you know,
I'll go to the cocktail party with you, Marion.
hey, I've got this, I've got a deal for you, and here's what it's gonna cost, you know? This is what you get.
You can run around Annapolis with me.
Lori or Phil will bring the booze, Lou will bring the cigars, and we're ready to go.
And Ralph is not far from us.
And that's a good point. You know, you've been to the presentation, you've seen what we've done and accomplished, and you see the direction we're going. So, you know, spread the good word, call your friends, let them know that there's an opening here and that we are holding on to a rocket that's going to take off, you know, spectacularly. The upside to this is tremendous for everybody, the investors and the patients and the like. So it's just a wonderful project to be involved with, and we're all very excited about the future here.
Oh,
I
yeah,
think,
that
go ahead.
part's easy.
Marion, you and I started a dialogue, I don't know, was it this morning, yesterday, about some connections. We're not going to let that go. And, you know, and what, you know, if you want answers to this question more specifically, let's talk about who it is and we'll give you the answer in a damn hurry. And, you know, you can see we have a pretty broad team of people who can do that. It isn't just your old buddy, you know, he's got around him an incredible team of people who are making this happen. And that's what's different from where we were a few years ago.
Well, I think it's very exciting, so. And it's
Thank you.
nice to meet everyone because not that I understand what it is, but I got the basics and that chart on the chart where it keeps getting better and the risks
Mhm.
get lower.
Ready?
Yes,
I like that. I could
I'm done.
understand
Wrapping
that.
up. I just said I have to jump.
good.
Okay, well, thank
Thank
you,
you.
Lori.
Thanks,
Um,
Lori.
Okay, we're all off.
Yeah, I
then.
think we probably ought to call it a day. We had an early one this morning. So Marion, thanks for being on, and for all the other people, some of you who I don't know, but thank you for being there. For those of you who have invested, besides Marion and Alex, who I see here at the moment, and Ayad, you know, keep in touch with us. And if you want to engage in further work with us to help us, just call us up. We are happy to take enthusiastic help, because
We will also be posting the recording of this, and we can provide the PowerPoint to anybody. If you've got somebody that wants to know more about it, we're happy to set up a meeting, or we're happy to send them our executive summary and our deck. So don't be shy. let us know.
Well, this worked, except I didn't get the email early enough. But this, to have a deadline for a meeting works, you know, a deadline's good.
I love deadlines.
You know, it makes people get out and hustle a little more, whether it's going to the plane or what. So,
Good, great point, Marion. Thank you very much. Tell your brother he's a bum.
oh, he's
Yeah.
there. He
He's
came
there?
up
Where
with
is he?
Kathy.
Oh, he's Kathy. Oh, George. Geez, I'm sorry.
Is he still there?
He hung up after I did that, I think. He's never gonna
Oh.
talk to me again. So he's also been an investor, and every time I go to New York, he hosts me. So he and his wife Kathy, provided my second home while we were doing all these presentations in New York. So thank God for them. So, and I'm going to tell you, working with Bill Workmeister has been extraordinary. He's providing a lot of advice, and I'm counting on Bill to be a greater part of this endeavor in the future. So thank you, Bill, for coming on, and for others who are listening. This has been a lot of fun. Thank you very much. And Steve, thank you for joining in. We miss having you back here, and if you want to engage more, let us know. We'd love you have you be part of it.
Right.
Yeah.
Thanks, everybody.
Presentation.
Good night, everybody. Thank you.
Bye.
All
Bye.
right.
Right.
Good night.
Bye, yeah, Steve. So long.
They're gonna weigh you.